The moment your heart stops, a clock starts. Your brain does not switch off. It goes quiet in about 30 seconds, with every cell in it still alive. And then, around 4 minutes in, a slow wave crosses the surface of your brain, and the cortex goes dark behind it. That wave has been recorded directly in dying human brains. It takes 4 minutes to arrive because of what your brain spends every second it is alive. Your brain is about 2% of your body weight, and at rest, it burns close to a fifth of the oxygen you breathe. It stores almost none of it. There is no tank. Blood brings oxygen and glucose in continuously. Neurons turn it into a molecule called ATP, the only energy a cell can actually spend, and they spend it as fast as they make it. So the instant blood pressure hits zero, the account is already empty. Consciousness usually goes within 10 to 20 seconds. Within about half a minute, the cortex, the folded outer sheet where thinking happens, goes electrically silent. On a monitor, that looks like the end. Every one of those neurons is still there, still whole. Studded along the membrane of each one sit millions of tiny protein pumps, and every pump burns ATP to push sodium out and pull potassium in. That pumping holds a voltage across the membrane, like a charged battery, and that voltage is what a neuron is. Firing a signal spends the charge. Holding it costs far less. So as the fuel runs down, the cortex stops talking to keep the batteries up. A flat trace at 30 seconds is a brain on austerity. And in that silence, some brains do something nobody predicted. In 2023, researchers published EEG recordings from 4 patients who died in intensive care with monitoring already running. 2 of the 4 produced a sudden burst of fast, tightly synchronized activity, gamma waves, concentrated toward the back of the brain, in a region often linked with conscious experience. It is worth being exact. That is a recording of electrical organization, not of anyone's experience. There were 4 patients, all already comatose, and 2 of them did nothing of the kind. A study called AWARE-2 followed 567 hospital cardiac arrests. 53 people survived, 28 could be interviewed, and 6 of those 28 described a lucid, structured experience they placed during the arrest. The same team recorded organized brain activity during CPR as late as an hour in. But nobody has yet shown that the patients whose EEG lit up were the same patients who came back with a story. A 2025 model called Neptune tried to explain these experiences as pure physiology: falling oxygen, rising carbon dioxide, a flood of neurotransmitters as the pumps give way. And it drew a published rebuttal within months. So the honest answer is that we do not know. What happens next, though, we can watch precisely. Somewhere between 1 and 5 minutes in, the pumps lose. ATP falls below what they need to run, and they stop. Not one at a time, but essentially together, across whole populations of neurons at once. The voltage each cell spent its entire life holding collapses. Potassium pours out. Sodium and calcium pour in. And that is what makes it a wave. All that potassium and glutamate dumped into the narrow space between cells lands on the neighboring neuron and pushes it over the same edge. That one collapses and dumps its own contents onto the next. The failure recruits. It travels at a few millimeters a minute, unbelievably slow for a brain that normally moves signals at meters per second. In 2018, a team led by Jens Dreyer recorded exactly this in 9 dying patients whose cortex was already wired with electrodes after catastrophic brain injury, and whose families had agreed to withdraw support. The electrodes caught the wave arriving at each one in turn, in order, like posts going down a line. The median start was 3.9 minutes after blood flow made its final drop. It is the closest thing medicine has to a picture of a brain dying, and it turns out to have a speed. Here is the part almost nobody expects. That wave is the last reversible stage. The neurons it has crossed are depolarized, swollen, and in serious trouble. But for a while, they are still structurally intact. Put oxygen back in time, and the pumps restart. The sodium goes back out, the potassium goes back in, the voltage rebuilds. And the wave of death is followed across the same tissue by a wave of resuscitation. In time means something specific. Researchers call it the commitment point, the moment the shortfall has gone on long enough that the cell will die whether or not blood ever comes back. It is not a reading on a clock. It moves. Cold tissue reaches it later because cold slows every reaction inside a cell, which is part of why people have been pulled from freezing water long past the famous 6 minutes and walked away. And in 2022, a Yale team ran a perfusion system called OrganX into pigs a full hour after cardiac arrest and restored circulation, along with working cells in the heart, liver, kidneys, and parts of the brain. They were careful about what they had not done. No organized brain activity, no consciousness, no animal brought back. 4 to 6 minutes was never a cliff. It is what the budget buys at body temperature. The same accounting is running everywhere else in you at once, at speeds set by appetite. In any cell that runs out, the sodium pumps stop. Sodium builds up, water follows it in, and the cell swells. Calcium floods in behind it and switches on enzymes meant to be used only carefully. Calpains that cut structural protein, phospholipases that attack membranes, endonucleases that cut DNA. Then the lysosomes, the cell's own recycling compartments, split open, and the cell begins to digest itself. A neuron, burning constantly, gets minutes. Heart muscle gets a few hours on ice. Liver, longer. A kidney can be cooled and transplanted the better part of a day later. Corneas keep for days. Those windows come from transplant practice, so they measure how long an organ can wait and still work in someone else, which is not the moment its cells die. But the ordering is real, and it is why a transplant team runs several clocks at once on one donor. A body goes at the rate each part was spending, which leaves medicine drawing a line across a gradient. One route is circulatory. Circulation has stopped and will not restart on its own, which in practice means watching and not touching for 5 minutes. That number came from evidence. In a large prospective study of planned withdrawal, the latest a heart ever restarted by itself was 4 minutes and 20 seconds. A separate route tests for the irreversible loss of all brain function. Neither claims every cell is dead. They claim the process is not coming back. And none of this is a set of instructions. If something about your own heart worries you, that is a conversation for a doctor. And hours later, the same missing molecule turns up in the most familiar place of all. Muscle works by grip. Myosin, the motor protein, reaches out, takes hold of the actin filament beside it, and pulls. To let go again, it needs one thing. A molecule of ATP has to arrive and bind to it. Not to pull, to release. When the ATP stops arriving, every myosin head that was mid-grip stays gripped. The muscle is not tightening. It simply cannot let go. That is rigor mortis, setting into the face at around 2 hours and complete through the body in 6 to 8. The last flicker of gamma, the wave crossing the cortex, the swelling cells, the muscle that cannot open. Those are not 4 separate items on a list. One molecule ran out, and everything after it is the same shortage arriving in each tissue at the speed that tissue was spending.